Hello everyone, in this video we are going to learn about respiratory system of insects. In insects, exchange of gases is exclusively carried out by the complex tracheal system. The tracheal system is invaginated from the integument in large number of columbola, protura, some chironomids and endoparasitic hymenopteran and dipteran larvae, and tracheal system is lacking, and integument serves the function of gaseous exchange. In insects, the blood or hemolymph does not play any role in transport of gases, and the blood cells or hemocytes are devoid of respiratory pigments like hemoglobin or hemocyanin, etc. The organs of respiration. The tracheal system is ectodermal in origin. The integumental invaginations develop repeatedly from either lateral side of segments, often second thoracic to the eighth abdominal, leading towards the formation of external openings or spiracles or stigmata and internal tubes of the trachea. Classification. Most of the insects, the tracheae communicate proximally to the exterior through the spiracles, while innervate the tissues of internal organs after being modified into the fine ends and the tracheus. Such type of tracheation constitutes the open tracheal. In the immature forms of aquatic insects, the spiracles remain closed and absorption of O2 from the surrounding water is carried out by the specific integument structure, the gills, etc., and then the system is called the closed tracheal. Holonucleus, open type, metanucleus, and epnucleus, closed type. Cayley and Hinton in 1947 have proposed much elaborated scheme of classification of the tracheal system on the basis of functional spiracles as follows. Polynucleus tracheal system. It represents the open tracheal system and can be divided into three subtypes, which are the holonucleus tracheal system, consisting of all 10 pairs of functional spiracles found in nymphs and adults of large number of hemimetabola. Then heminucleus tracheal system, in which only 8 pairs of spiracles, the mesothoracic and first 7 abdominal spiracles are functional, whereas other pairs are non-functional, mostly in the larva. Then perinucleus tracheal system, in which 9 pairs of spiracles are functional, while only a single pair located on the metathorax is non-functional, as found in terrestrial larvae of Neuroptera, Mecoptera, Lepidoptera, Symphyta, etc. So different types. Polynucleus tracheal system, it represents the open tracheal system and can be divided into three subtypes, like holonucleus tracheal system, consisting of all 10 pairs of functional spiracles found in nymphs and adults of large number of hemimetabola. Then heminucleus tracheal system, in which only 8 pairs of spiracles, the mesothoracic and first 7 abdominal spiracles, are functional, whereas other pairs are non-functional, mostly in the larvae. Then perinucleus tracheal system, in which 9 pairs of spiracles are functional, while only a single pair located on the metathorax is non-functional, as found in terrestrial larvae of Neuroptera, Mecoptera, Lepidoptera, and Symphyta. Then oligonucleus tracheal system, it represents partial open tracheal system. It is further divided into three subtypes, like aphenucleus tracheal system, in which only 2 pairs of spiracles, first thoracic and last abdominal, are functional, as found in the larvae of Diptera. Then metanucleus tracheal system is that where only a single pair of spiracles, the last abdominal functional, as larvae of the last abdominal is functional, as seen in larvae of Culcidae, Tipulidae, Hypoderma, first larval instar of Cyclorrhapha, and aquatic Coleoptera larvae. Then pronucleus tracheal system is recognized when only a single pair of spiracle, the first pair of mesothorax, is open. Pupae of Diptera. Apneus tracheal system. It represents the closed respiratory system. All pairs of spiracles are closed and non-functional. Oxygen is absorbed by skin or gills as found in various aquatic and endoparasitic larvae. Then hypomneustic tracheal system. It represents partially degenerated or atypical tracheal system in which one or two pairs of spiracles disappear completely, and insect therefore bears often less than 10 pairs of spiracles as found in Malophaga and Sipungulata, 1 pair of thoracic and 6 pairs of abdominal. Thysanoptera, 2 pairs of thoracic and 2 pairs of abdominal. Hemiptera, Sterorinca, Coccidia, Columbola, Smithurids. Hyperneutic tracheal system. It shows presence of more than standard number 10 pairs of spiracles. Diplura like Gepias bear 4 pairs on the thorax and 7 on abdomen, in total 11 pairs of spiracles. Then anatomical organization. The tracheal system is composed of two structures, the internal tubular trachea and the external orifice, the spiracles. They are initially developed segmentally but later on fused intersegmentally, forming a complex tracheal system. It consists of the longitudinal trunks, transverse commissures, intercalated air sacs, and terminal fine tracheoles. The wall of tracheae shows similar but reversely organized structure of the body wall consisting external basement membrane, middle epithelium, and inner cuticular inchima. The cuticular inchima is divided into series of rings forming a spiral coil or filament trachea, except the tracheal region where it is found as penidium. The penidium prevents collapsing of the tracheal wall. Then tracheal branches, trunks and commissures. Tracheal branches, trunks and commissures. In Epterichota, except Lepismatidae, the tracheae are long, unbranched and free tubules, while in other insects they undergo enormous branching and unification. The proximal most part of tracheae, which runs for a short distance from spiracle, is called the spiracular trachea. It gives off 3 branches, anterior, posterior and visceral, constituting dorsal, ventral and visceral tracheae respectively. The spiracular tracheae give out lateral branches which fuse with alike branches of neighboring segments and form lateral longitudinal trunks. Similarly, fusion of dorsal, ventral and visceral tracheae results into the formation of dorsal, ventral and visceral longitudinal trunks respectively. In each segment, these paired trunks are transversely connected with each other by the tracheal commissures. In air sacs at various places, the trunks and commissures dilate into large air spaces which undergo a periodic contraction and expansion in order to accelerate ventilation and to provide buoyancy during flight, and also function as heat insulators, sound resonators, or hydrostatic organs in aquatic insects. They are developed abundantly in flies, wasps, bees, ants, dragonflies, and scarabei beetles. So dorsal longitudinal trunk, heart, dorsal diaphragm, visceral trachea, dorsal trachea, spiracles, cuticular coil, ctenidium, tracheal lumen, tracheal region, visceral trachea, gut region, lumen, ventral trachea, ventral diaphragm, ventral longitudinal trunk, nerve cord, ventral commissure, longitudinal trunk, spiracular trachea. All these are the structural parts of a tracheal system, trachea and segmental tracheation. Okay. Next is about spiracles. They represent simply the orifices or openings of internal tracheae. In almost all insects except Diptera, the spiracles are wanting from the prothorax. There are only 10 pairs of spiracles, 1 on each meso and metathorax and abdominal 1 to 8 segments, situated laterally, often primarily on the pleura on either side. They vary widely in their structure and can be classified into following types. Simple spiracles. They are tubular and open directly from exterior to the lumen of trachea, for example Thysanura. Then aeriate spiracles, atriate spiracles. Simple atriate. Spiracles open first into the cavity or tubular chamber, the atrium, containing sieve plate or hairs, and with stigmatic glands. It bears external opening called the atrial orifice, which may be simple or provided with a valve. The internal opening is called the tracheal orifice. The closing or occluding apparatus develops as the external closing apparatus associated with the atrial orifice in the thoracic atriatus spiracles, and as the internal closing apparatus associated with the tracheal aperture in the abdominal atriatus spiracles. They are found in various pterygota. Then bichambered spiracles. The spiracular cavity is divided into two chambers, anterior and posterior. The posterior chamber is called the occluding chamber and it bears internal closing apparatus. They are represented by abdominal spiracles of the ants. Then biferous spiracles. Spiracles bear two external openings which are secondarily developed while the primary atrial orifice is closed. They occur in the larvae of Coleoptera. Okay, then multiferous spiracles. The spiracles have more than two openings found in the third instar larvae of Cyclorhiza dictera. In Glossina and Etrus, spiracles open to exterior by about 500 pores developed in the chitinous plate and they remain open due to the absence of closing apparatus. So these are some different type of spiracles. Simple spiracles, atriate spiracles, bichambered spiracles, biferous spiracles, multiferous spiracles. Yeah, that's it. Simple, atriate, bichambered, biferous and multiferous. So we learned about the organs of respiration. So I'm going to put a done mark here. Then mechanism of gaseous exchange, next topic. The gaseous exchange along tracheal system can be divided into two parts: transport along trachea and transport along tracheoles. The transport along trachea, air tube diffusion. Among most of the terrestrial insects, oxygen enters the tracheal system either through all holoneustic or some of the spiracles, mostly by diffusion mechanism called the air tube diffusion. Diffusion mechanism is simple in small insects like fleas, while in others the spiracles are provided with the opening and closing apparatus that controls the rate of diffusion. The diffusion control lies under the control of segmental respiratory or sympathetic nerves, which innervate the spiracles. O2 CO2 tension in the blood and CO2 content of air is taken in. The spiracles remain opened when the CO2 tension is raised. The ventilation as a major supplementary mechanism occurs in higher insects, which initiates sufficient uptake of O2 into the trachea and circulates it through the tracheal system to various parts of the body. In higher insects, ventilation continuously forces the gas to flow in the anterior-posterior direction through the longitudinal trunk, the pumping action of the abdomen. The pumping action is derived from the elasticity of the body wall and the muscular action of the abdominal region, columnar dorsiventral flattening. It is initiated by telescopic action in case of Hymenoptera, Diptera, Orchestra, Coleoptera, and steady static compressed action is found in Diptera. Okay. So alternative movements of compression and expansion pump air out from into the tracheal system through the spiracles. Air may flow in and out of the spiracles, and such movement of air is called tidal. The next respiratory cycle will be the pumping action of abdomen acts mechanically on the spiracula valve and on the volume of tracheae. As a result, the airflow through spiracles appears to be a cyclic phenomenon. The respiratory cycle is thus divided into three phases: the inspiratory phase, the compensatory phase, and the expiratory phase. The inspiratory phase is comparatively shorter than the remaining two phases. In grasshoppers, the inspiratory phase lasts for about 1/4 second, while the compensatory as well as respiratory phase each appears for 1 second. Collapsing of tracheal trunk and air sacs forces air out of the tracheal system, while its subsequent expansion sucks air in again. The spiracles are closed for most of the time for respiratory cycle, as it is essential to avoid loss of water, and thus they are opened only to supply a sufficient amount of O2 to the system. Not all spiracles take part in inspiratory or expiratory phases but are specialized for one of them. In the low-caste Schistocerca, spiracles 1, 2, and 4 carry inspiration, and spiracles 10 conduct expiration in normal condition, but spiracles 5 to 10 serve during more active expiration. Inspiratory spiracles open for 20% of the cycle, while expiratory spiracles open for 5 to 10% of the cycle. The compensatory phase therefore lasts for 70 to 75% of the cycle. In honeybee, apis spiracles 1 and all abdominal spiracles conduct inspiration, while the spiracle 2 carries expiration. What is ventilation? In faster flying insects, abdominal ventilation is supplemented by the pterothoracic ventilation, P is silent, and the third abdominal ganglion acts as a pacemaker. Abdominal ventilation supplies in Schistocerca about 150 liters per air kilogram per hour, while the pterothoracic ventilation provides about 350 liters air per kilogram per hour, as CO2 demand of flight muscles increases 400 times during the active. Cyclic release of CO2 occurs commonly in diaphosin pupae, where about 90% of CO2 is retained in tissues. In Hylophora pupae, post-stream bursts of CO2 may be of 7 hours. It occurs in some diaphosin larvae and pupae of Lepidoptera, Coleoptera, and locust nymphs. During such conditions, CO2 is absorbed in the hemolymph and converted into the bicarbonate. The transport along tracheoles, tissue diffusion. Each tracheole ends blindly, and the blind end of tracheole is made up of a single cell. All tracheoles are filled with the fluid at the blind end. The tracheal fluid is not an ultrafiltrate or hemolymph, but it resembles the amniotic fluid, having similar composition as that of a molting fluid. It is a watery fluid rich in protein components but without chloride. It is replaced by active transport or absorption but not by osmosis. Wigglesworth's hypothesis. The fluid contents are absorbed by the wall of tracheoles and oxygen enters the system. Removal of fluid from endings take place during the activity with a deficient oxygen supply. This removal is brought about primarily by physical forces produced by the products of metabolism, which are not immediately removed or oxidized at the site of activity. As the air enters the tracheal system, the fluid is absorbed into the tissues by a mechanism of active transport due to secretory pressure. The tracheal fluid is never withdrawn from the tracheal system but varies in degree of withdrawal into the tissue with the need for oxygen. It was earlier suggested that the osmotic pressure controls the level of tracheal fluid and opposing forces maintain the balance. It is, however, discarded now as it is proved that capillary process in tracheoles operates against the swelling process of plasma. The difference in permeability between the outer and inner layer of the tracheole is a very important factor in the absorption mechanism. Permeability constant of CO2 in tissue is 36 times greater than that of O2, so that despite its higher molecular weight, CO2 travels more quickly than O2 through the tissues, and it therefore brings an adequate supply of O2 in the tissues and also suffices to take the CO2 away. Next is about aquatic respiratory organs. The aquatic insects possess closed respiratory system in which the spiracles are closed, and especially modified integumentary structures, the gills and hair, assist the system in obtaining the oxygen from the surrounding water media. These structures vary in their topography, morphology, or mode of action and are said to be developed independently in different groups of insects. They are described by various authors, but still available information on their physiology is fragmentary in various respects. They can be grouped into following types, like the abdominal gills. They occur as the lateral segmental integumentary outgrowths in the form of folate or lamellate epimera nymphs or filamentous Plecoptera, Zygoptera nymphs and cruciform larvae of Trichoptera gills. They are richly supplied internally with trachea. They occur primarily or 57 abdominal segments are formed. In all, 7 pairs of lateral abdominal gills in most of the aquatic forms. Epimeroptera, Zygoptera. While secondarily, they may develop in the form of finger-shaped or copious tufts on various parts of the body of Plecoptera nymphs like submentum, neck, thorax, coccyx, first 2 or 3 abdominal segments, or on caudal abdominal region. In Trichopteran cruciform larvae and pupae, the gill filaments are arranged in dorsal, lateral, and ventral rows on either side of the abdomen, while these comodioid larvae are devoid of gills and integument carries the relevant function. The gills are often the free external structures, while in some Ephemeropteran nymphs they are concealed in the branchial chamber formed from the lateral LD walls pertaining to the thoracic and first 7 abdominal segments. The abdominal gills are undeveloped in newly hatched nymphs and occur in only few species at the adult stage. Besides these groups of insects among Coleoptera, aquatic larvae of Hygroilia genus bear the paired filamentous gills ranging from 6 to 10 pairs. The Hygroia, there are 3 pairs of gills near bases of legs and remaining 3 pairs on the first 3 abdominal segments. In other Coleoptera, they are confined to the abdominal segments.